Marielle. Welcome everyone. For those of you that it might be your first Salish Sea Roundtable, just a little reminder that this is sort of a co-managed group between DFO and Marielle's team down at the University of Washington, and some others who we are actively recruiting participants to come and do presentations on things that are relevant to our Salish Sea. So we've had a number of different really interesting talks over the last few months, and if you're interested in giving a presentation, feel free to reach out to either of us. So we'll just kick it off. I'll just introduce our three speakers today. So I'm joining you from the Pacific Science Enterprise Center, which is in West Vancouver, on the home of the traditional territories of the Tsleil-Waututh, Squamish, and Musqueam. We had a really interesting archaeological report here with some fun facts that this place used to be referred to as Stoopdale, which is loosely translated as stinky place. And that was from the abundant salmon river that was from just behind our property here. So I'm joining you from here, as is Steve Healy. I will let the other two introduce themselves and where they're coming from shortly. So we have three folks joining to co-present today. We've got Steve Healy, who's a biologist with DFO in Science Branch and located here at PSEC with me. We've got Anne-Marie Norris, who's also a biologist with DFO in the Ecosystems Management Branch. And then we've got Healy Crozier, who is our lead natural resource technician from the Tsleil-Waututh Nation in the Treaties, Lands, and Resources Department. So a very interesting collaboration with three of them delivering today. So with that, I'll just hand it over.
Thanks, Cher. Thanks for the introduction and for inviting us to speak today. We're excited to share a bit about the work that we've been doing together. And so Steve, but also Healy and I are both also joining you today from the unceded ancestral territories of the Tsleil-Waututh, Squamish, and Musqueam nations. I'd say we're all really grateful to the nations and their ancestors for their long-standing stewardship of these lands, and privileged to be able to continue some of this work in the waters in and around Burrard Inlet. Steve and I also just wanted to extend our appreciation to the Tsleil-Waututh field crew before we get going here. We spent countless hours learning, laughing, and collaborating with them, and the work we're going to discuss today really wouldn't be possible without their partnership. You can go to the next slide, Steve. So just a bit of an outline showing the structure of today's talk. We're going to start with a bit of background on Tsleil-Waututh Nation, talk about the Burrard Inlet Environmental Science and Stewardship Reconciliation Agreement, or BIESSA, which is really the basis for this partnership. And then we'll talk about some of the collaborations that we currently have underway, and end with some final thoughts on working together through this reconciliation agreement. Just so you're aware, we will be jumping around presenters a little bit. I'll start off with the first half of the slides, pass it to Steve to finish off, and Healy is going to weave through with the Tsleil-Waututh context throughout the entire presentation. So you can go to the next slide, and I will pass it to Healy now to talk about an overview of Tsleil-Waututh Nation and situate us within Burrard Inlet.
Hi everyone, my name is Healy Crozier. I'm going to give you some of the Tsleil-Waututh context. So Tsleil-Waututh Nation are known as people of the inlet. On the map on the right there, that shows the Tsleil-Waututh consultation area, but kind of in the very middle between the district of North Vancouver and Burnaby. That area is Burrard Inlet, which we're going to be talking about a lot today. So historically, Tsleil-Waututh have lived along the shores of Burrard Inlet for thousands of years. And historically, they had a population of around 10,000 individuals, down to unfortunately just a few dozen due to contact and the disease that came with it. The current population sits around 700. Historically, Tsleil-Waututh moved with the seasons and the available resources, with the villages being occupied all along the shoreline and waterways. The three main village sites on Burrard Inlet were Wauwauchin, Tumtuatum, and in Tsleil-Waututh. And they really fished, hunted, and gathered all throughout the territory, and specifically relied super heavily on marine resources for about 90% of their diet. Stewardship responsibilities have really been maintained through time, and that work continues today, and that includes practicing sustainable harvesting. You can go to the next slide, Steve. So now I'll talk a bit about the Treaty Lands and Resources Department and the field crew. So TLR, so Treaty Lands and Resources, was formed in the early 90s, and it was formed to bring together existing fisheries work the nation was doing, ongoing treaty negotiations, and upcoming land and resources work. Tsleil-Waututh is not a treaty nation. Those negotiations fell apart and have not continued since. The department now has about 60 full-time employees, including the field crew, which is made up of 7 community members and myself. A little bit of background on the field crew and the work we do. So our field crew isn't actually a formal Guardian program, but we do conduct a lot of activities you would see in a Guardian program. Our main role, though, is really data collection, stewardship, and monitoring work. The map on the left there is kind of a more zoomed in map of Burrard Inlet. All the boats show the different areas we work. So there's the Outer Vancouver Harbor, Inner Vancouver Harbor, Port Moody Arm, Indian Arm, and the Indian River Watershed is where we conduct pretty much all of our fisheries work. You can go to the next slide, Steve. So next I'll talk about the Burrard Inlet Action Plan. So in 2017, BEEP, as we'll call it, was completed, which created a vision for environmental stewardship in the inlet that really guides a lot of the work our field crew does, as well as the department in general. It also guides the work of the Burrard Inlet Environmental Science Secretariat. So the main goals of this plan relate to water quality, different habitat types, key food populations, cultural continuity, and governance, I should say. And right now there's actually an update coming to BEEP, so that should be coming out within the next year or so, and that will update Tsleil-Waututh's vision and priorities moving forward into the future. And I'll hand it off again now.
Yeah, so the Burrard Inlet Environmental Science and Stewardship Agreement is really the vehicle for the partnership that we'll be talking about today between Fisheries and Oceans Canada and Tsleil-Waututh Nation. This is a nation-to-nation reconciliation agreement that's intended to foster coordination and collaboration between the Tsleil-Waututh Nation and Canada. The agreement itself was signed in August of 2021. The signatories include Fisheries and Oceans Canada, Environment Canada, Transport Canada, Crown Indigenous Relations and Northern Affairs Canada, and the Tsleil-Waututh Nation. And as part of the agreement, there was a requirement to form a secretariat, which includes members from all the signatories, and that secretariat is required to meet at least twice a year to coordinate and collaborate on science and stewardship work they have going on in the inlet. And typically these meetings will include the signatories to the agreement, but occasionally we do bring in guests who have relevant overlapping work in the area. The agreement also includes consistent funding over the 10 years of the agreement to Tsleil-Waututh for their work on science and stewardship. And you can see the image there on the left side. That's the area, the coordination area covered under this agreement. And it's similar to the map you saw from Healy as the area that the field crew do a lot of their work in. And then, of course, we also include the freshwater tributaries to this system. And a lot of the work that you'll see is being done up in the Indian River Watershed. Can go to the next slide. So we have 3 collaborative projects that we're going to touch on today. We'll start with a Dungeness crab acoustic telemetry pilot project, then talk about our clam beach research, and finally end off talking about the work that's going on in the Indian River Watershed. Next slide. So starting with crab telemetry, I'm going to pass it to Healy to talk about the Tsleil-Waututh context around crabs.
Thanks, Emery. So historically, as I said before, Tsleil-Waututh's diet was primarily marine-based, with crabs being an especially easily accessible resource as village sites were found right along the shoreline. Today, crabs are actually the only easily harvestable marine resource for Tsleil-Waututh community members. So from April through August, community members are down on Maplewood Mudflats, which is right in front of the community as shown on the map there. And people typically are harvesting crabs by hand, either by just walking the intertidal or by snorkeling and collecting crabs as they go. You can go next slide, Steve. Some community members also use traps to do this trapping work or crabbing work, but most of them don't actually have access to boats, so most people go by the hand picking method. And in the last 2 years, the nation has tried to increase the number of members actually getting out and harvesting. So they've had knowledge sharing events where they ask current harvesters to go out on the flats and share their techniques with anyone from the community who's interested in crabbing, and really to get a next generation of harvesters going. Additionally, the nation has also been conducting contamination analysis on crab tissue, but unfortunately, we still don't know exactly how representative a crab is of an area because there's very limited data on crab movement and home ranges, which Anne-Marie will talk a bit about next.
Yeah, so as Healy was just mentioning in the last slide, there's not a lot of understanding on movements of crab around the inlet. It's been coming up as a topic both from the nation as being of interest as well as from DFO. We thought maybe we could put some tags on some Dungeness crabs and see if we could figure out what key regions there might be in the area, or if there's habitat connectivity, or just generally answer some questions about crab movements. And the Burrard Inlet is an intensively fished area for crabs commercially, recreationally, and for food, social, and ceremonial purposes. So it seemed like it might be useful to just try to get a better understanding of the crabs and how they're using the area, especially since we don't really understand the connectivity. At the same time, we're using this pilot to develop capacity for future acoustic tracking work in Burrard Inlet for both DFO and the nation. The pictures there, you can see the top picture, someone is helping tag a crab. The picture at the bottom gives you an idea of what a tagged crab actually looks like. So we use epoxy to affix the tag and include a small message indicating that if someone should catch the crab, they should release it back to the water. You can go to the next slide. So we've been tracking crabs in 2024. We're doing it again this year in 2025. Last year, we tagged and released a total of 17 male crabs in 3 different locations around the inlet. All of the crabs were actively tracked. So that's using a pole with a hydrophone attached, which you can see in the picture there. The person at the front of the boat is spinning the hydrophone in the water. And then I'm sitting listening for pings on the VR100 to see if we can locate a crab. Each crab has a unique frequency that's pinging, so we know which crab we're chasing down. We've also done some range testing to see what decibel readings we need to get to be within 20 or 30 meters of a crab. And the higher the decibel reading, the closer we are to the crab. So we do our best to get as high as possible, but it does depend a bit on weather and how noisy the inlet is on a given day. And then in 2025, so this year, we tagged 30 crabs. We focused more on one area this year. So that's the area around the Maplewood Mudflats, directly in front of the Tsleil-Waututh community that you just saw in Healy's previous slide. And I will show another map in a second. And as she indicated, the area is used extensively by the nation for food, social, and ceremonial crabbing. It's also a key part of the commercial and recreational fishery in the area. This year, we modified our method slightly using a blend of passive tracking, which requires receivers in the water that record the decibel levels of tags that pass by, in addition to active tracking that requires us to be out on the boat looking for crabs with the hydrophone. And this year, we also tagged some females as well as males. We do try to choose crabs whose shell condition indicate that they've recently molted. And then the hope is that they don't molt again until March or April. That gives us a good chunk of the year where the tag should still be affixed to their shell. You can go to the next slide. So this is a map of the area that we're working in. To the right there, that situates you in Burrard Inlet. The middle picture, you can see where the passive receivers are located. So the receivers are either attached to a sediment or a cement patio block that you can see in the top left photo there, and then placed on the ocean floor subtidally, or they've been hung off of permanent buoys. And then the crabs that are being passively tracked talk to the receivers that they pass by. So we have a spread of receivers up both sides of the inlet from Second Narrows to Waututh Park. And hopefully we can do some comparisons this year of the active versus passive tracking techniques. You can go to the next slide. So we're still really in the data gathering phase of this project. We don't have a lot in the way of results to share, but here are some movement maps from 2024 at 2 of the 3 locations where crabs were released. Each one of these colors on the map is an individual crab. The one on the left is near the Indian River estuary. The one on the so the Maplewood Mudflats area, which is where we're focusing this year. So So the data aren't cleaned up or anything, but it's just an idea of some of the movements that hopefully we'll be able to use to determine distances traveled, movement rates, or maybe home ranges. You can see some areas where clearly crabs are congregating a bit more. There's always a crab that goes on a very long journey from where the rest of the crabs are, but there's a lot more work to be done here. This is just really the preliminary stages. You can go to the next slide. Yeah, so we're going to leave crabs behind and move into the clam beach research. And again, I'll pass it to you, Healy, to talk about clams in the Tsleil-Waututh context.
So some background on clams in Tsleil-Waututh territory. So historically, bivalves were one of the 4 main food resources for Tsleil-Waututh Nation. Again, villages were all along the inlet, quite often with clam beds right at their shores. So in 1972, the clam fishery was closed on Burrard Inlet due to contamination from industrial development. In 2001, Tsleil-Waututh began working with Environment and Climate Change Canada towards opening a limited clam harvest. In 2012, the nation began conducting small-scale clam surveys, really just to see what species were where. In 2016, the nation had the first successful opening of a limited clam harvest in Burrard Inlet. This was literally one beach, a couple days, and that's it. And pretty much every year since 2016, there has been a harvest at that location, other than 1 year where the nation voluntarily chose to close it due to a rainfall we weren't expecting, essentially. And then in 2021, there was a Masters of Science project conducted by one of our coworkers, specifically studying clam species distribution and abundance. You can go to the next slide, Steve. So here's just a photo from about 2012. And again, the nation was really just getting out there to see what species are on what beaches on the inlet. And I'll hand it over again to Anne-Marie.
Yeah, so as Healy was saying, Tsleil-Waututh has been conducting clam surveys across the inlet annually since 2012. And then in 2023, DFO, so Steve and I, started working alongside the nation to build out this survey and see if together we could pair the population data with some environmental data and build off some of the previous master's research that they had done. So the overall objective of this work is to monitor environmental and physical factors at some key clam beaches in Burrard Inlet and gain a better understanding of factors that could be influencing clam productivity and invasive species distribution. And hopefully this research can help provide some guidance for enhancing or restoring clam habitat, either through clam gardens or perhaps through remediation or offsetting projects in the future. You can go to the next slide. So this is a map showing the 12 clam beach locations where we've been working. As you can see, there was some aim here to get spatial coverage and also align with beaches that were of importance to the nation. The sites range from Second Narrows Bridge up the Indian Arm, and there's one site up the Port Moody Arm. And all of these locations vary in their characteristics like slope and sediment composition. You can go to the next slide. So this is a schematic of what a beach might look like with respect to data loggers. Not every beach has all of these components, but just to run through an example, here we have temperature loggers on the surface in the low, mid, and high tide zones, as well as a key logger in the mid tide zone that sits just below the surface, capturing more accurately what temperature clams might be exposed to. There's also a temperature logger up in a tree measuring the ambient air temperature. And then there's a subtidal mooring, which sits on the ocean floor. Similar to the crab receiver picture you saw, the loggers are attached to a cement patio stone. And there we have current meter, salinity, and a temperature logger. In addition, we took sediment samples at each of the beaches to look at potential metal contamination, as well as some key nutrients. And then the population surveys were done with standardized quadrats across the low, mid, and high tidal zones at each of these beaches. Next slide. Just some nice fieldwork pictures. There's the one on the left there. You can see Healy in the water as we're pulling up our mooring to download the data. And then some clams about to be weighed, some sediment about to be processed, and one of the field crew measuring a clam. I'm going to pass it to Steve now to talk about some of the preliminary findings with the clam data and then carry on into the Indian River Watershed work.
All right, so thanks. I get the coveted role here of going through some of the data analyses and kind of preliminary results from some of the clam work here. And so We are sort of terming this sort of emerging trends. So we're sort of late in the analyses here with a lot of this clam beach work, but we're still sort of actively kind of working with these data. So hence why we're sort of calling this emerging trends or preliminary results kind of thing. So one thing that was immediately sort of jumping out was the association between clams and beach slope. So that's what you're looking at here, these two regression lines. And so beach slope is on the x-axis, and that's in a negative degrees. We took the reading from the top high tide zone down to the low tide zone. So another way to think of this is beaches are points further to the left along the x-axis, or sort of a steeper beach relative to ones further towards the right, or more of a flat or level kind of beach. And so if you do this similar regression against biomass of everything at these beaches, you see a positive relationship between beach slope and biomass. But then we can break it up by invasive species density and native species density, which is what you're seeing here on the y-axis. So invasive species on the log scale on the top, and native species density on the log scale on the bottom. And so we see a positive relationship between invasive species and beach slope. So higher densities of invasives at those sort of lower slope beaches, maybe trending towards a similar relationship with native species, although not significant. And so we could go through obviously our whole data set and do a whole bunch of these types of regressions and look for significant results. But a more appropriate way, of course, would be to sort of use models that can encapsulate a bunch of these variables all together. And so we've been running a bunch of generalized linear models and generalized linear mixed models. And so that's a bit of what you're looking at here on the right. So this is a partial effects plot of a number of different models that we've run. So you can see the different responses at the top here. So we've run models where biomass is the response for everything. And then we split it up by invasive species and native species in terms of their density. Basically, the way to understand this plot is that the circles you see here in the middle, I don't know if you guys can see my cursor or not, but those are the estimated effects of that variable, which you see along the y-axis. The lines sort of connected to that are the error on that estimate. And if they don't cross this middle dashed vertical line at 0, then that sort of implies that it's a statistically significant effect. So what we've kind of highlighted here, we blanked everything else out, but we're just looking at tidal zone, and we're starting to see a tidal zone stratification of natives versus invasives. And so we see the top panel here is tidal zone for the low, so down towards the bottom part of the beach. And then below that is the high tide zone. And for the purposes of this figure, the mid-tide was acting as kind of our reference site. So this is sort of the effect at the low relative to the mid, and the effect at the high relative to the mid. So hopefully that kind of makes sense. But what we're basically seeing is that at the low zone, native species much more positively associated with that low zone, and we see the opposite trend with invasives. So they're negatively associated with the low zone. And when we go up from the low zone up into the high stratum, then we see actually kind of the inverse relationship start to happen. We see native species much more negatively associated or sort of marginally negatively associated with the high zone. And maybe not a statistically significant result, but invasives tending towards a more of a positive effect. And so we can kind of look at that as a partial effect, but we can also turn this into a predicted sort of way. So this is sort of an output of the model. So you're looking at the same sort of things here. This 3-panel figure on the left, we have biomass of all species. In the middle is invasive density, and the right panel is native density. The 3 bins along each plot are for the high, mid, and low tide zones. And so if we just look at this figure on the left here, this is the predicted value in grams per 0.125 meters squared is just per quadrat. And so we see as you go from high down to the low tidal zones, we start to see an increase in biomass of clams that we're detecting or we would predict at those zones. And this, again, is sort of taking all of our variables into consideration and essentially sort of holding those other variables constant. And then when we jump over into this middle panel and the right panel, which is now the units are in the number of clams per quadrat, we see for the invasives, which is the panel in the middle, going from high to low, we start to see less and less invasive clams. And then we see again the opposite trend with the natives. From high to low, we actually see more native clams. And so this kind of gives an idea of why we're sort of splitting things up and looking at biomass of all species and then splitting it by invasives and natives. It tells us different things about the population structure at each beach. So biomass might give us an idea of sort of like the total productivity or potential productivity at one of those beaches. Whereas then if we break it up by invasives and natives, it starts to tell a bit of a different story, whereby if we just look at biomass, it tells us that, there's, higher biomass at the low zones, but that's likely driven by a larger proportion of these native species. And even though they're at quite lower abundances, you'll note that these two axes of the invasives versus natives are the natives are an order of magnitude essentially lower. But those tend to be much larger individuals. So larger butter clams, horse clams, even like native littlenecks are quite a bit bigger than some of the invasive species that we see on those beaches. And these trends are sort of what we would expect sort of biologically based on the literature and what we're seeing at these beaches. So it kind of gives us a bit of a check that our models are doing what we hope for them to be doing in accurately describing these beaches. So that was sort of a bit of a stratification at the tidal zone, but we also see a separation spatially. So when we regress natives versus invasives density and vice versa, we see overall the negative association between the two. So native species tend to be negatively associated with invasive species and vice versa. And this is probably best encapsulated with this very bottom panel you see for latitude, whereby native species negatively associated with latitude and invasive species positively associated with latitude. And probably the best way to describe this is native species we find in much higher densities in the sites to the south, closer to the city core, relative to the invasives, which are found in higher densities up north towards like the Indian River and some of those northern sites that you've seen on the map earlier. Kind of the opposite of what you'd expect, given what we might think for things like invasive species and introductions through anthropogenic causes and stuff. And so we're trying to sort of determine what's kind of driving this effect, because obviously latitude itself is not really an effect. This is likely just acting as a proxy for some other physical or biological factor that varies across that gradient. And so there's probably a few things going on here which we're trying to tease apart, but one of which we think is playing a pretty strong role is the effect of temperature, which we'll touch on shortly. If we jump into some of the sediment and nutrient characteristics from our sediment samples, I've just highlighted sand, which is just a percentage of sand in the sediment. And then this variable nutrients you see just below that, which is a bit of a collapsed variable. Actually, we had quite a strong multicollinearity among a bunch of variables in our nutrient signal. And so this is a bit of a collapsed variable that includes bicarbonate, phosphate, ammonium, and then a bit of the metals signature as well. And you might notice, while sticking with the nutrients, that native species were the only group that kind of jumps out as being impacted by nutrients. And this probably is largely driven by the bicarbonate and phosphate signal from those samples, but still sort of teasing some of that apart. And then similarly, when you look at the proportion of sand, we see native species positively associated with sand and invasive species not as much, or maybe slightly marginally impacted by sand percentage in the sediment. And again, where we're wanting to go with a lot of this work is to put this into sort of predictive terms. And so this is, again, what you're looking at here on the x-axis is just the percent sediment, or percent sand in the sediment rather, on the x-axis, and then on the y is the predicted native density. And so as we increase sediment sand percentage, we predict that we would see more native species density. Not huge numbers more. We're not sort of like, multiplying our native species by a lot by going from say 40 to 80, but an increase nonetheless. And then similarly, I didn't show sort of the GLM output effects plot of this, but this is sort of a similar predictive plot of temperature on the x-axis versus invasive species on the y. And so this is from the data loggers that Emery was talking about a minute ago, specifically the subsurface data logger that was in the clam habitat at about 20 centimeters under the surface. And so what you're seeing here obviously is a pretty clear relationship between the peak temperature that clams experience at those beaches and what we predict the density of invasive species will be on those beaches. As we get into those high temperatures, there's pretty big error bars on that, obviously, but again, a positive relationship. And so this kind of all gives a little bit of an idea of sort of like where we're going with these data and sort of how we're interpreting this. But there's certainly more to come, and we'll be likely writing this up into a bit of a peer-reviewed paper quite soon. So we're going to jump from the marine now, and we're going to freshwater to talk about some of the Indian River watershed work that we're doing with the nation. So I think I'm passing it back to Healy now.
Awesome. Thanks, Steve. So some background on the Indian River watershed. So the watershed is considered by many Tsleil-Waututh members to really be the heart of Tsleil-Waututh territory. It's really one of the only wild watersheds left in the Lower Mainland. I just want to add that it has no dam, no hatchery. There's Forest Service roads and a couple transmission lines. But again, really the heart of the territory, as acknowledged by a lot of community members. The Tsleil-Waututh village site that I mentioned earlier is located in the estuary and was historically the home base for salmon harvesting in the watershed. The nation has been conducting fisheries stewardship work in the watershed since the late 1980s, starting with spawning channel enhancement with DFO and then stock assessment work. In the 90s, the Aboriginal Fisheries Strategy Program was implemented with DFO, and stewardship work was further expanded from just those spawner counts and enhancement work to then include juvenile minnow trapping surveys, side channel habitat assessments, and other work that was needed to get that stuff done, essentially. So in 2023, we began our environmental monitoring work with Steve and Anne-Marie, and this included the installation of temperature and depth loggers, a rainfall gauge, a suspended sediment load sampling device, and most recently, dissolved oxygen loggers. You can go next slide, Steve. So the reason we started to look at using dissolved oxygen loggers was that in 2023, on a massive pink run year, because we only get them every other 2 years here, we actually saw the main stem Indian River go dry in 2 sections, each measuring over 100 meters in length. This was about 6 kilometers from the estuary, and there was still about another 5 kilometers of spawning habitat upstream. So this was actually only the 3rd time since the 1980s this had happened in the watershed. And when it happened, we had about 100,000 pink salmon stuck within 2 kilometers downstream of those dry out sections that couldn't make it to their spawning grounds. At the time, we were super lucky to have one of the contractors we really trusted having an excavator already up in the watershed. And because of our strong relationship with the DFO restoration unit, we were actually able to get to work within a week of noticing the problem to really begin working on remedying it. Unfortunately, during this event, we did witness some pretty massive die-offs. This year, we saw the return of the offspring of that run. Luckily, it doesn't seem to have had a huge impact on this year's run. When we got to work, the excavation work took about 3 days to complete, and that allowed us to reestablish access to those more northern spawning grounds. And while this event was like absolutely terrifying to see, you never want to see it, a sort of silver lining that came from it was really this opportunity to work together to better understand the factors that are causing these drought conditions. So that's why we ended up installing DO loggers as well, was because the die-offs and just to understand what's going on. And if you can flip to the next slide, Steve. This here is just interesting. This is looking essentially downriver. So in the first photo on the left there, you can see like the river just goes dry. That's the main stem. For 100 meters there, it was dry. And then the photo on the right shows when we were done. And you can see there's all those shadows. There are pinks that have now made it up that section and are able to make it towards their spawning grounds. So with that, we're going to now focus on some of the DO logger data. I'll hand it back to Steve.
Yeah. And so just to kind of echo what Healy just said there, so what sort of turned out to be a has turned out to be not sort of a very good situation for the watershed or for the fish, has kind of given us a little bit of an opportunity to use this to study dissolved oxygen in the context of salmon densities and water levels, and sort of what sort of conditions lead to these hypoxic or potential hypoxic events in a coastal system. And again, sort of the hope with this work is that through studying this, understanding sort of these trends, gives us the understanding of when we might need to think about mitigation or intervention to sort of minimize any impacts to fish. So in the last 2 years, we've been out installing dissolved oxygen loggers in the watershed. And so this is just kind of a bit of a map showing where some of those sites are. And you can see the river sort of flows from top of screen down to the bottom. And the red circle here shows, this is just like a Google Earth image showing actually where the dry up is. You can see the river and the main stem go subsurface there. I believe this imagery was from 2023, just before the mitigation work took place, although I'm not totally sure. And so this is a sort of general area of concern. And so we've deployed DO loggers, which you can see in this sort of inset image here. This black long pipe has a depth logger, which is paired with each DO logger. And then this little shorter pipe here attached to it is the dissolved oxygen logger. And so each site has both depth data and hourly dissolved oxygen. We deployed one site above, one site below that dry up area, and then two rather in side channel rearing and spawning kind of habitat to sort of look at things like potential groundwater recharge of DO in that area. Also, this site you can see at the farthest left here, this number 18 site, when fish obviously weren't able to go up past that dry up zone, they really stacked into that sort of, there's a bit of a, quite a bit of spawning habitat in this sort of alluvial fan here. And then this map off to the right here is just showing further down in the watershed. So if you can see these two sites at the very top here, you see 18 and 24, which correspond to the two on the left here, just sort of around that dry up area. And these are two sites further down, one of which we chose based on some of our temperature data, showing it as one of the hotter sites in the watershed where you might expect to see DO levels drop in the summer. And then this site really far down was one that we chose from local knowledge from some of the field crew who identified potential issues with DO even sort of on a normal year. So we wanted to focus on that site as well. And so here we are out installing some of our gear. There's Anne-Marie and Healy looking happy after we've just installed. This was just earlier this year. You can see, hopefully you can see the black DO and depth pipe just right here attached to a rock. And we've got a paired staff gauge so we can look at water depths at that site as well. And then on the right, you'll see, on the back here, the very far back, that's the exact same site, that same logger and staff gauge. And this is later in the year in about, I believe, early September. And hopefully you can kind of see this dark, dark water here. This is just a whole bunch of pink salmon milling in and around that data logger. And actually just upstream of this site this year is a place that we were sort of actively watching, and Tsleil-Waututh crews were out actively watching water levels because they were getting to sort of a concerning level for pinks to get by. And I think just wanted to highlight with this slide that this really just shows how we can work together to pair data sets. So we're pairing the spawner abundance of the Tsleil-Waututh field crew who are out there at least a couple times a week, getting spawner abundance by reach and by habitat type. And that's pairing it with our various data loggers deployed throughout the system. And so just to kind of give a little bit of a flavor, we haven't really worked these data up yet. We just pulled these loggers about a month and a half ago, and still are sort of compiling all of the spawner abundance data and all that sort of stuff. But this is just sort of raw data you're looking at here. I'll just walk you through the figure. So the x-axis is time or date. So it goes from about June until mid-October. Along the y-axis is dissolved oxygen in milligrams per liter. This sort of opposing y-axis on the right, I'll pull those data in shortly, but this is sort of an approximate or assumed pink salmon spawner abundance. Again, we don't have those data yet, but I'll sort of show roughly what the sort of abundance distribution for pink will look like in the system. And then we've got a couple lines for both years of data. So this bottom line here is for 2024, and this sort of slightly higher top line is for 2025. And then we've got this sort of dashed line at about 3 milligrams per liter, which we're kind of calling an acute stress. It's, you know, an approximate threshold whereby sort of below that or roughly around that level, we might start to see concerning levels of dissolved oxygen for salmon that would lead to sort of more acute stress, mortality, and that sort of thing. Obviously, even above that, we might start seeing sublethal effects and those sorts of things, but it's sort of more a visual representation to give you an idea of when DO levels might start causing adverse impacts to salmon. So I'm going to basically walk this figure forward now in through the rest of the fall. You'll see 2024 more or less stays the same. There's a bit of a bump up here that was likely a rain event. And that's one thing that we don't have on this figure is all the depth data, which we'll be sort of pairing with these as well and looking at, you know, when we see big pulses of water, typically we'll see a bit of a bump in DO as well. And that's kind of what's happening in these little spikes here. You see on the 2025 data, which largely, unfortunately, are decreasing and more or less actually kind of bottom out just before or around early October. You'll see down here to the right of the figure, getting below sort of that acute stress line. I guess anyone maybe who knows anything about pink salmon in this area will know that 2024 and 25, the difference between those is that in 2025 is when we actually had pink salmon in the watershed. So we've kind of got almost a pink salmon effect that we can kind of use as a bit of a, you know, 2024 is kind of acting as a bit of a control year that we can make comparisons against. And so we're trying to understand basically, you know, the effect of things like water levels and pulses of rain. We've got rainfall gauge out in the watershed as well. And sort of what, or how much of an effect this biomass and the drops in water levels are having on our dissolved oxygen profiles at all of our sites in the watershed. So just a bit of a teaser of kind of what those data look like thus far. And another sort of just visual way to look at this, we also deployed some time-lapse cameras in the watershed. And so this image is from July of this year. You can kind of see this little inset here in the bottom left is trying to show roughly where that camera is facing. So this little triangle cone is sort of giving the field of view of the camera looking sort of northwards, just upstream of where we saw the dry up event before. We had a camera placed right at the area that dried up in 2023 as well. But as I mentioned before, this was kind of the area that was a bit more of concern this year and people were keeping a bit of an eye on. So here it is in July 2025. Water levels already looking fairly low. Again, this is kind of the main stem of the river. But if we advance that forward into what things looked like in September, this is kind of peak migration for pink at this point. And I'll just draw your attention to a few things that might not be super clear on your monitors. Again, I'm not sure if you can totally see my cursor here, but about middle, close to the top here, you'll note that, you know, that's the entire main stem of the river, probably only a meter and a half wide. So not a huge amount of water coming down the watershed at this period of the year or time of the year. You might notice this dark band kind of up through this bottom part of the main stem here. That is a whole bunch of pink salmon, again, trying to wait, or sort of waiting and milling to try to get up this, what amounts to a sort of small riffle. And then you might notice a bunch of happy crows and gulls and a bunch of dead salmon carcasses along the shore, and they're happily feasting on those. So anyone who's been to a salmon spawning system in the fall will know that, you know, this type of thing is not uncommon. Salmon will spawn and then die and complete their life cycle, of course, but the Tsleil-Waututh field crew were out there quite regularly cutting open the bellies of females and noting that a huge proportion of them were pre-spawn mortality. And so obviously it's a bit more of a concern when, or it is a concern when fish are unable to actually spawn before they die. And so don't want to sort of leave this on a negative note, but we're hoping really that our work that we're doing and pairing data sets will give us more information to be able to understand when, you know, these sort of mitigation or intervention type things might need to be taken into consideration. And then with that, I'll pass it back to Healy.
Okay, so something a bit more positive, I guess. and in the theme of really pulling together our data sets. So I'm going to give a little bit of a teaser as to something we have on the go right now. So I've been working on a report with Stephen Emery's help to pull together our 2024 juvenile minnow trapping data from the Indian River Watershed with some of our in-stream temperature data for 15 sites within the watershed. So we were essentially looking for any significant relationships between catch per unit effort, or CPUE, and temperature, and then looking for relationships between fork length and temperature. You can go next slide, Steve. So significant relationships were observed between water temperature and larger coho and larger chum lengths in the month of March, as well as between coho catch per unit effort and water temperature. So larger chum and coho body sizes in March could be due to increased food availability and ideal thermal conditions in some of those warmer sites that allows for quicker juvenile salmonid growth. Additionally, the higher catch per unit effort of coho at some of these warmer sites is likely indicative of greater food availability at those sites, which therefore increases the capacity of those locations to really support more juvenile salmonids. And then finally, we found that the highest coho CPUE was at a main stem site that is pretty much completely dominated by naturally occurring large woody debris structures. So it's likely that this high density of coho at these sites is due to the scouring created by that large woody debris and the cover and refugia offered by that large woody debris. It also may increase food availability due to stuff coming off those structures. This information specifically was really kind of exciting to see as we're starting to consider doing large woody debris work in the main stem of the Indian River. The nation's done a lot of side channel restoration, again, since like the 80s, and we're kind of at a point where we're like, what do we do next? And like so many of the other rivers on the coast, the watershed is really lacking large woody debris due to historic logging, and it will not have those inputs for 80 to 100 years because there aren't trees that big. So we're really hoping to take this route of going for the installation of large woody debris structures, but it's quite a long-term process. So it will be a lot of work and a lot of time before we get there. But another interesting thing with it is it'll loop into Tsleil-Waututh's Indian River Watershed Integrated Stewardship Plan. Part of the goals in that includes reinstalling, or I should say, installing and increasing large woody debris abundance in the watershed. And in addition, with the drought impacts, installing the large woody debris structures could really help restore hydrology, and in addition to improving salmonid habitat. Next slide, Steve.
And I'm just, while you're, oh great, you're on final thoughts. Perfect, Healy.
Perfect. So final sort of thoughts here. So again, our success here is really coming from our relationship that we are lucky to have. We're working together, we're pairing complementary data sets, which we're using to better identify and study complex ecological systems in what is a really changing climate. The benefits of BIESSA for Tsleil-Waututh include stable long-term funding for science and stewardship. It allows for flexibility to fund emerging priorities, such as the DO loggers to monitor drought conditions and the impacts of that. And it really creates a forum for coordination and collaboration with an area of a lot of complicated overlapping jurisdiction. And I guess at that point, if there's time for questions, we can go to that.
Yeah, perfect. Thank you, Healy, Anne-Marie, and Steve. Just a great presentation with so many interesting components that you guys are working on. You know, climate change to food security to salmonid survival. Just an amazing snapshot of that. So I appreciate how you were trying to sort of give a broad overview, but it's difficult to get into a ton of detail on a particular project because they've got so many. So I'll just open the floor up for any questions. People can put their hands up or post it in the chat. We'll try and manage both of those. We have only about 10 minutes left because the presentation was so great with so much interesting work on there. So while those are just rolling in, I had a question on the aquatic invasive species side of things. So you guys are looking at the different clam species. Is there interest to still consuming those invasive clams, like in terms of still it being a food source? Like I know it's not the preferred or traditional, but just any interest in that, and how does that fit into their Burrard Inlet action plan?
I guess I can answer that. So right now that the limited harvest that Tsleil-Waututh does, that's been ongoing since 2016, it is specifically a harvest for invasive softshell Myonaria clams, because there are not high enough abundances to harvest the native species. So that's essentially what the harvest has been, and there's a lot of ongoing work one day to hopefully get to more of a native clam harvest, but right now the priority is on those invasives.
Nice. Perfect. Thanks, Healy. Any other questions from around the room? I know there's a lot to digest, and we can sort of parse out who that it might go to. Looks like we've got a question from Amanda in the chat. So regarding bivalves from the 70s closure to the conditional opening, what did collaboration look like with for those of you that don't know all the acronyms, it's the Canadian Shellfish Sanitation Program run by Environment Climate Change Canada, CSSP ECCC, which is a big mouthful, in order to address contamination. I know their historical practices with CSSP is once it's closed, it's infrequent that they have the resources to go back and look at reopening it. So just any comments on that for Amanda and others?
I guess I can answer this one too. So yeah, as you said, typically when an area gets closed, it's closed. So this was Tsleil-Waututh really taking the lead and working with ECCC. So essentially what had to happen was, I believe I said starting in 2005, that work began. So from 2005 until 2016, 4 times a year, the field crew would go out and collect surface water samples at around 15 to 20 sites on Burrard Inlet that were then sent to ECCC for testing to look for fecal coliform levels. So that went on, and there had to be a set number of data points that were considered clean for the harvest to open. So there's that portion of it. And then in addition to that, there was also ongoing proxy sampling. So essentially near the harvest beach, there are mussels hanging in a crab trap that kind of represent the clams on the beach because they're filtering the same water. And bi-weekly through the fall months and weekly in the summer, after they've been at that site for 2 weeks, our crew goes out, collects, there's like 30 of them. They're in prepackaged little things that we set up and hang there. We take those, they get sent to the lab, and they get tested again. So that was going on year round. So that's kind of like the step with the actual shellfish. And then right before the harvest, it has to be within a week before, we go to the actual beach where we're going to harvest, and we dig up 30 clams, so soft shells that we're going to be eating. And then those get the final test to make sure that the clams on the beach are actually safe for consumption. That's kind of the whole entire process. But I believe if there's 2 negative hits through our sampling in the year, the whole entire harvest is canceled. So
yeah, they've got some pretty strict protocols associated with, and for good reason. We also don't want people getting ill or dying from consuming shellfish. So they definitely have their reasons there, but way to go to continue to pursue them. So our next question from Ellen, and I apologize if I'm mispronouncing your name, but some questions with respect to the crab telemetry. Wondering if they're sort of moving as a bit of a population. You've got individual trackers on individual species, but are you seeing it sort of move as a population? And do you see any primary differences between males and females?
We haven't really got there yet. So like Anne-Marie said, we're sort of still actively collecting those data. We didn't actually tag females last year. So we've only really in the last sort of few months been starting to track females. But we do see, again, like you can kind of look at those map figures that were sort of shared, like there do seem to be areas of sort of aggregations, but we still sort of need to dive into the data in a fulsome way to sort of better understand that. We want to sort of create sort of home range maps and almost sort of heat maps of where we're seeing higher aggregations of crabs.
Right.
Did that answer the question?
I think as good as they're going to get with the information that you guys have.
Stay tuned.
Yeah, perfect. Some other questions from Amanda related to the the contamination that, like, you know, was the source of the contaminants removed in order to move through on the shellfish sanitation? Like, you know, you're talking about coliform, fecal coliform. Was there point sources that were removed, or what was the likely source of that, or just general water quality has improved over time? Any comments on that?
All of Burrard Inlet was closed at once, so the whole entire thing. There was not primary contamination sources as far as I'm aware. They're identified and addressed. Burrard Inlet is quite large, and our harvest beach is not very close to the industrial activity, we'll put it that way. So just in a general cleaner portion of the inlet, I guess you could say.
Yeah, for those of you not familiar with the area,
blanket closures are applied. Yeah.
Yeah,
blanket closure for the whole thing. They won't go in and retest. So yeah, that's why it's a limited harvest for one specific area.
Yeah, and for your beach, right? So yeah, no, that's it's a complex question. Okay, any other questions? It was a great presentation. Got to learn a lot about the program, and I think just a great example of collaboration between a nation and DFO. Like the science team is doing wonderful stuff. I've gotten to work with Steve and Anne-Marie on a couple projects, and there's some others that have joined in on the call today that have worked on various projects with Tsleil-Waututh as well. So really great to see those collaborations and partnerships happening. So we appreciate you guys all having the time to bring that forward to this group. This group is super interested in things going on in and around the Salish Sea. So this is a very topical presentation. And I'm sure you guys wouldn't mind if there was any follow-up questions if they got directed to you. So yeah, I think, Marielle, any final plug for our next one? If not, I'll just say a big thank you to the 3 presenters today, and then for Marielle and her team for running the back end of Zoom, which I think capable of operating. So thank you for all of that support and help, and much appreciated. So maybe I'll let you get
I just threw up our emails there too if anyone needs them. I forgot to flash them earlier. So there they are. I think you can see my screen now, can you?
Yes, we can. And we'll make sure that those get into the slides too that get shared out. So thank you very much for being open to those conversations.